Hello, my name is Tristan Arroyo, and I will be presenting West Nile virus. So, the clinical importance of West Nile virus is that it possesses great health risks on a global scale. One of the main things is that it can cause major outbreaks like COVID-19, what we see now, and that West Nile virus also causes severe disease symptoms in humans. And one of the main reasons it's commonly researched is that there are no current treatment options for humans for the disease symptoms that are present. And I feel like understanding the West Nile virus on a more molecular level will possibly illuminate targets for vaccine development or antiviral development. So, a quick introduction into West Nile virus. Its family is Flaviviridae. Its genus is a flavivirus. Its genome consists of positive single-stranded RNA. It can cause acute or chronic infection depending on what symptoms are present. It is neurotropic. Since viruses are seasonal, West Nile virus is usually seen in the summer through fall. And West Nile virus is commonly found in Africa, Europe, the Middle East, North America, and West Asia. So, quick about transmission. So the human infection is often a result of mosquito bites, and the mosquitoes usually obtain the infection from birds. So birds are the reservoir and are also known as amplifiers because the virus can build up in great numbers within the bird's system, and then the mosquito will then bite the bird and pass it on to, I guess, what dead-end hosts are, which are either a horse or humans, which are the most common dead-end hosts. So they're considered a dead-end host because they present the symptoms and don't really spread the virus past that. However, there are other forms of getting the virus, like exposure in a lab, blood transfusion, organ donation, or from mother to child via like breastfeeding, pregnancy, or birth. So the two common symptoms of West Nile virus are either West Nile fever or West Nile neuroinvasive disease. The fever is, I guess, less serious. 1 in 5 people will present with the fever, and you usually get like flu-like symptoms or a bit more serious flu-like symptoms. While the neuroinvasive disease is a lot more life-threatening because you present with encephalitis, meningitis, or meningeal encephalitis. So, and the neuroinvasive is actually seen in 1 out of 150 people. And even though that seems like rather small, in 2019, 907 cases were reported to the CDC, and 66% of those were neuroinvasive disease. So we can actually get into the genomic structure or organization of West Nile virus. So the West Nile virus consists of a icosahedral nucleocapsid, which is contained in a lipid bilayered envelope. So so the genome is translated into one single polyprotein and is cleaved by host and viral proteases into 3 structural proteins and 7 non-structural proteins. So your structural proteins are C, PRM/M, and E. So the C is for the capsid, the E is to help form the envelope, and the M is for the membrane. And you either have PRM or M depending on what stage virus is in. So during early development stages, or you would have a PRM, and towards the assembly, like the final assembly of the virion, you would have an M protein, and then you would have then the 7 non-structural proteins that are necessary for replication and assembly are NS1, NS2A and B, NS3, NS4A and B, and NS5. So here we have a linearized picture of the genome, and you can see where the proteases will cleave and the organization of the proteins on the genome. So the viral genome actually has a cap at the 5' end, and it lacks a polya tail at the 3' end. So it has a single open reading frame, which is franked by 2 UTRs, one at the 5' end and one at the 3' end. The 5' end UTR functions as a promoter for the polymerase, and the 3' end is important because it contains a complementary sequence to the 5' end, which allows for the genome to circularize, which is necessary to enter replication. And the UTR also has these RNA or resistant RNA structures that are there to help, protect from degrading the RNA by the host exonucleases. Um, Even though those resistant RNAs are still there, the genome does get, I guess, degraded a bit by the exonucleases, and this results in a sfRNA forming. And this sfRNA is actually quite important to the virus. I guess research papers showed that it helps stimulate the virus and with its like pathogenicity and um, inhibition of immune response. So it kind of has a positive effect or a positive interaction on the virus with the host. So we can get into a bit of the interaction of the virus with the host. So the virus enters the cell or its host via endocytosis, and once it enters, it is transferred, I guess, into the cytoplasm, where it then releases the viral genome, and the nucleocapsid disassembles. However, the mechanism or the way the nucleocapsid disassembles is not fully understood. But what is understood is that, or I guess kind of proposed, is that there is a ubiquitination of the C protein on the nucleocapsid, and that kind of assists in the disassembly of the, I guess, nucleocapsid. So once the viral genome is released, it is then transported to the endoplasmic reticulum, where the initial translation and replication occur. So this initial translation is quite important because it is a prerequisite for replication to start, because the virus needs its proteins to, I guess, assist in regulate the replication cycle or process. Um, so here you have like I said, the three structural proteins get translated, and those are really needed until assembly is, I guess, ready to begin. However, the non-structural proteins here are rather important. Two of the real important ones are NS3 and NS5. NS3 is there as a protease, and it also works or functions as a helicase, where NS5 is the RNA-dependent RNA polymerase, which is the polymerase for the virus. And it also is a methyltransferase. And the other five non-structural proteins participate in replication and assembly. So one of the interesting things, or the reason why I put this figure here on the side, is that I guess West Nile virus, or I guess flaviviruses in general, the ones that do contain a cap can either go through a cap-dependent in translation or initiation, transition initiation, like eukaryotic, I guess eukaryotic translation. So it's the same way, or it can like not go through that same process. Depending, I did read somewhere where it said that depending on the levels of initiation, eukaryotic initiation factor 4E, which is important for the binding to the 5-cap, is levels are high or low, it will either use the eukaryotic, or I guess 5-cap dependent way, or it won't. So if it does use the 5-cap dependent way, it will pretty much go through the entire same process as translation initiation, like forming the pre-initiation complex and everything, like how the eukaryotic, I guess, cells go, or cells processes. And it kind of also competes with the normal hosts for all of these initiation factors. So, and then once these proteins have been translated, they then kind of go in to help replication. And one of the main ones is the polymerase, so the NS5. So the NS5 copies the RNA, I guess the original genome RNA template, and uses that as a template to synthesize negative RNA, which is then used again to to form multiple copies of positive RNA. And these positive RNAs are then used as for translation to make proteins, or are used to package into new virions. And that's pretty much the importance of the replication process, is pretty much just to make a lot more of the positive RNA in the end, to either get more viral proteins or to make more RNA in order for the virions to have it enough. So I guess one of the conclusions is that understanding a bit more of what the I guess proteins, the viral proteins, are used for, and how they are important for the gene expression of the West Nile virus can lead to a treatment option in the future, and possibly one of these proteins can be targeted for vaccine development, and you will see a lot less uh, possibly death or from the actual symptoms that people present with. So here are my references. And one of the questions I wanted to pose for anyone who watches a video is to, I guess, come up with a potential mechanism for how the ubiquitination assists in the degradation of the capsid and to release the viral protein.